From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 43, recorded on September 25th, 2023. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hi, Vincent. Hi, Tim. How you guys?
And from New York, Tim Chung.
Hello. Hi, everyone. Here in New York, absolutely miserable weather.
Oh, no.
Yeah, we have this hurricane going up the coast. It's been raining all weekend.
Yeah, it's
still raining.
I had to come into work, into the lab on the weekend, and I normally bike. And the one thing that is worse than working in the lab over the weekend is working in the lab over the weekend with soggy, wet socks.
Yeah. Sorry
Absolutely
about that.
terrible. But yeah.
All right. We have some paper for you today. Recently, we've been talking about hallucinogens, and today we're going to move to analgesics. Tim is going to shepherd us through. Hey, how about that? I got both Jason and Tim in. Tim's going to shepherd us through this paper. There you go, Tim.
Yeah. When I was telling my lab mate that we're going to do this paper, he's like, "Oh, are you guys just moving from one illicit substance to the next?" The answer is yes. We are doing that. So today's paper, let me pull it up. Deals with opioids. So the title of the paper is Coordinated Activity of a Central Pathway Drives Associative Opioid Analgesic Tolerance. So that's a lot of mumbo jumbo that we're going to decode. It's published in Science Advances, so that is the open access version of Science. So the paper would be open access for everyone to look at. And the two co-first author, Yiwen Hu and Guicheng Zhao. Last author, senior author is Wei Xiong, and I really apologize for my pronunciation. I'm from Hong Kong. I speak Cantonese. My Mandarin is, as you can tell, terrible.
Oh my gosh. Didn't know it was that different.
It's completely different. Oh, it's different. People get offended when I can't speak Mandarin. And also people get offended when I speak English in this country with a funny accent. So I have to deal with two versions of Chinese and two versions of English. It's tough life.
Sorry.
Anyway, so these authors are, are mainly from China, from Anhui Province Key Laboratory of Medical Imaging Aging Research in Hefei, China. So this paper deals with one kind of thing that's been on the news for quite a few years now in America is the opioid epidemic. So this, if I remember correctly from reading the news, is severe enough to maybe contribute to the average life expectancy of Americans actually going down. So this is a pretty severe problem. And as people know, it is originally driven by pharmaceutical company kind of downplaying the addictive effects of opioids. Now, opioids is prescribed for a very good reason, which is a very, very effective painkiller. It has analgesic properties, so pain killing, pain numbing. But it also can cause a host of other side effects. So, for example, it can cause users to feel euphoric, so feel very good, and that's one of the reasons why it has potential to be abused. Those two are fine, but the third one is the one that kills you, which is it causes breathing suppression. So in large enough dose, you just stop breathing. And that is one of the reasons why the mortality rate has really spiked up. And it's actually relevant to NYU here, where I'm at, because we actually had a few institutes previously that was named after the Sackler family. And since the opioid epidemic story has come out, all of them no longer are called Sackler Institutes. They have all been renamed. Still, that's, scant consolation to people who's lost family members to this epidemic. But yeah, so opioid drugs has a host of several effects. And it causes, side effects such as breathing suppression, as we mentioned, breathing suppression, and also constipation, which is an odd one. I didn't know this until I was very surprised when I found out that one of the drugs that you can take when you have GI problems, two-bucket disease and diarrhea and all that, one of the drugs you can take is called Imodium, and that's actually opioid receptor agonist. So it actually activates your mu opioid receptors, but only in the gut. It doesn't go into your brain. That's why it doesn't have abuse potential, at least in normal doses. So how does opioid work? So it's a little bit of background here. So how do opioids work? Opioids, our body naturally produce opioid peptides. So opioid peptides are kind of a small length kind of peptides made of amino acid that are genetically encoded by our DNA. And they are released by a bunch of cells in the body, including neurons in the central nervous system. And they're released presynaptically, and then they bind to postsynaptic opioid receptors. So this is how, all these neurotransmission works, is something gets released, it crosses the synapse, and then it binds to a postsynaptic receptor. And there are several classes of opioid receptor, but the main one we're going to focus on today is one called mu opioid receptor, which is critical in the analgesic, so the pain-killing effect of opioids. So it has been very well known that if a person is taking opioid for a long time, then the same dose of opioid becomes less and less and less effective. So this is an effect called tolerance. And in a way, it is your body's kind of way of trying to maintain some sort of homeostatic stability. So, for example, if I have a very high elevated level of opioid in my system repeatedly for a very long time, your body kind of thinks, oh no, my set point has now been changed, so I better make myself less sensitive to this elevated level of opioid, so that in the future, when there's a further increase in opiate peptide level, I can still detect it. So this is kind of your body's way of trying to maintain things in a so-called dynamic range that you can still detect things.
And this is very common for neurotransmitter systems, because if you think about it, synaptic transmission has to be working almost perfectly all the time, despite all the changes that can happen, especially in the brain when you have plasticity at synapses. So there's lots of homeostatic mechanisms that want to keep your signaling in that window, that sort of physiological window. But I think as you're going to get to here, the simple idea was that receptors would just change their levels depending on how much activation they would get. So a simple homeostatic loop here was that too much activation, you would get either desensitization of the receptor or internalization of the receptor, and that's why you would need more neurotransmitter, or in this case the agonist, the drug, to get the same effect. But it seems that that's not the case. It's been unclear why you get tolerance or how this works.
It might be the case, but it's not the only case. It's not the only contributing factor. So it's possible that the kind of so-called receptor desensitization still occurs, but something else might contribute. But But I forgot to mention, you might be wondering, why do our bodies have these opioid receptors and these opioid peptides? Why are we making it? Is it just so that we can all go and take morphines and all these opioid substances? As it turns out, our body would. Well, actually, I don't know whether we know why our bodies make all these opioids peptides, but the thinking is that in our daily lives, there are often times when we do things that are in pain, that causes pain, and we do want ways of numbing it so that we can get on with our lives. So, for example, especially in times of like when we're very stressed, you might want to ignore some of the pain. So it might be the body's way of, at the beginning you sense there's a pain, but then you can increase your endogenous opioid peptide level. For example, there's been found that if you exercise, if you go running, your endogenous opioid peptide can increase its transmission so that you don't feel as the little nagging injury you have in your knee might be a little bit less painful. So that
in
might
other
be
words,
one of
an endorphin is an opioid.
Exactly. So exactly, endorphin, endogenous morphine is one of the body's naturally producing opioid. But since your body is used to sensing and working with your own body's natural opioid, it makes sense for it to tune up and down its sensitivity. But when you hit it kind of with a sledgehammer with drugs to really manage pain, that is the occasion when your body might have a little bit of difficulties dealing with. Now, as Jason alluded to, when you give opioid for a very long time, a synthetic opioid, so also natural opiate drugs for a long time, you develop tolerance, so you are less sensitive to the analgesic effect of the drug. So it works less well. Basically, the drug stops working, and what people do is they escalate the dose. And this is problematic because if you escalate the dose too much, the one thing that you can get is breathing suppression and death. So people used to think that it is due to your receptor being internalized, or maybe the signaling mechanism gets desensitized, get downplayed so that it cannot signal as well. But in the 1980s, a long time, half a century ago, researchers found that some of these tolerance to opiate are actually learned in a similar way as to Pavlov's dog learned to associate ringing of a bell with a piece of steak showing up and therefore salivation. So what these researchers did was they injected morphine into rats repeatedly for several days in a very distinct context. So you do it across, and then they can test how well this morphine blunts pain using an assay where they put the rat, or in our case, in the paper we're going to talk about, it will be mice, but you put the rodent on a hot plate, and this hot plate is turned up to about 50 Celsius, so a little bit hotter than in a hot spring. And what a normal mouse would do is you put them on a hot plate and they go, ow, 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 and they jump around. And when that happens, you remove the mouse so that they don't, or the rat, so that they don't burn. And you note down how long it takes for the mouse to go, ow, 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 it's too hot, it's too hot. If you inject the rodent with morphine, they would quite happily sit there on the hot plate for quite a while. So in this study, they have a maximum duration of 30 seconds. So after 30 seconds, if the rodents still don't go, ow, 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 they would take that rodent out so that it doesn't burn their paws. So this is a way of assaying how effective morphine's analgesic effect is. So if you inject the rat for, let's say, 5 days with morphine, on the first day, the rat would happily sit on the hot plate for all 30 seconds. But day after day, you would start seeing the rat start showing in an effect and start going, ow, 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 it's hot, earlier and earlier. So by day 5, the morphine's effect is much, much lower, and the rat is showing pain again. But what's interesting is that the same rat, if you take the same rat that now is showing morphine pain again, so the morphine doesn't work anymore. The same rat, if you inject the morphine and put the rat in a new context, and then test the rat on the hot plate, the morphine now works again. So it looks like what is going on is that the rat was associating the development of the tolerance to morphine was associated with the original distinctive context. And if there's a shift in context, then the morphine suddenly stopped working again. And this actually has potential health consequence because, as it turns out, when researchers went back to some of the records of people who died of overdoses, a significant portion of those people were taking the opiate, usually it's heroin, in a new context. So the thinking is that these people were previously using their opioid drugs in a familiar context, developed tolerance to the opioid drug, but for whatever reason, they go to a new context, and the same dose of drug now suddenly has a much greater effect, not only for pain numbing, but also for breathing suppression. So this is obviously yes.
Let's put it in the context of Jimi Hendrix or Janis Joplin, right?
Yeah.
Who both died of heroin overdoses. So what would be the different? They're just in a different place when they overdosed. Is that enough to be a different context?
I think that would be the argument. Did Jimi Hendrix die in London? Wow, this might be.
I think so. Yeah,
this might explain it.
I think so.
But did he? I need to look up what he died of. I don't know whether it's from heroin abuse or from something else, because I think he choked on his vomit.
I guess the question here is, I mean, maybe do we know that the breathing effects are mediated by the same central nervous system receptors, or is it peripheral?
That is one interesting question. We don't know. They seem to correlate together. I do not know whether we just don't know. Yeah.
I think,
at least I don't know. People might know, but I don't know.
It's hard to imagine. So the central nervous system signaling it seems, would make sense that you could modulate it top down. The brain is controlling that effect, but the breathing effects you would expect maybe would be much harder to change.
But I think you can, in some sense, breathing can be controlled by your central nervous system, by the brain. Like I can voluntarily withhold my breath.
Yeah, it is controllable, I guess. But anyway, I think it's a very interesting idea.
It is. But actually, there's in the paper, they actually showed that this context-associated tolerance can also develop for breathing. So that suggests a central mechanism.
Yeah,
as they said, like a brain mechanism.
So Jimi Hendrix died of barbiturates, but Janis Joplin died of a heroin overdose. So
I see. Yep, both with abuse potential, both suppress your breathing.
Yeah,
not sure if barbiturate has a context learned tolerance.
Well, I think this contextual aspect of things is certainly true for drugs of abuse like cocaine and methamphetamine, because there's sort of this learned association cues where you go into relapse just because you've made that association where it's more conscious, or it's more like, I'm craving drug because I'm in a context that's familiar. So there's this idea of sort of learning that's sort of gone wrong is not new, but I think it's really interesting that it's so powerful.
Yeah. It's also a little bit, let's say, annoying, because it's not under your conscious control. You're kind of subjected to the environment you're in, which kind of tells us how much we are actually just a function of our environment. So it is interesting that your environment, if you repeatedly pair opiate drug with a specific context, you can develop this tolerance that can be attributed paired with the environment. So this, the purpose of this paper is to ask what goes on in the brain when this is happening, and can we maybe manipulate the brain to either to stop it from happening. So the first experiment that these authors did was they first replicated the finding that we've been talking about. So they had one group of mice that received daily injection of morphine, kind of systemically, so injected into the stomach for 5 days. And for the experimental group, these mice received the morphine injection in a very distinct context. So it's kind of like a square box with like bright barcode stripes. So the mice are really seeing, I'm in a new place. And also I'm feeling kind of nice and on morphine when I'm in this context. And then they also, every single day, they also put the mice on this hot plate to see how strong the analgesic effect of morphine is. So that's the experimental group. The control group is quite interesting. You would expect the control group may be the same. You might expect the mice in the control group might also be having morphine and then putting in this new context, and then after 5 days put to another distinct context that's different and show that suddenly morphine now works. But if we're interested in what is going on during those 5 days when we're giving repeated morphine, which is what we're trying to do here, these two groups would actually receive the same treatment. And it's not actually until the very last day that the difference between the group is revealed when you put them suddenly, one of the group in suddenly a new context. So instead, what these researchers did was, for the control group, they injected still the same amount of morphine, 5 days of morphine, but instead of putting the mice in a specific distinct cage, a new context cage, they put the mice back home in the home cage. And it's also been previously shown that if you inject morphine to a mouse or rat and then just put them back in the home cage for 5 days, you develop the morphine tolerance that you develop is less strong than if the context is very distinct. So it's there's still a slight, you still see a reduction of morphine's effect, actually, even in the mice who receive these morphine in the home cage. And that speaks to potentially receptor desensitization that we're talking about here. So if you give morphine every single day for 5 days, your receptors are also working to downregulate so that you basically turn down your sensitivity to morphine. But if the mice are given the morphine only in a very distinctive context, this tolerance is much more rapid. It's actually worth quickly talking about what might be going on in the home cage. Why are these mice, the tolerance level, the development of tolerance slower? And why don't these mice develop this context-associated tolerance? Because they're still getting morphine in the home cage, and it's still the same home cage day after day. What probably is happening is that since morphine only works in the system for maybe 3-4 hours, these mice would have the analgesic of morphine effects paired with home cage, but then every single day the effect goes away. So they have a chance of, they have a opportunity for the system to essentially extinguish what people call. So it's kind of like if you're Ivan Pavlov and you're ringing a bell and giving your dog a piece of steak, the dog would salivate, but then, afterwards, if you keep ringing the bell but don't give the steak to the dog, the salivation eventually decreases. So maybe the mice in the home cage learn a little bit every single day, but at the end, because they're still in the home cage, when this effect goes away, then they unlearn it or they have an extinction effect. Whereas in the experimental group, the mice are always getting morphine in a distinct home cage, so they don't extinguish this tolerance response. So these researchers show that in this experimental group, where you give morphine in this distinctive new context, the tolerance, development of tolerance of morphine's effect develop very, very quickly. And what's interesting is that we know that more receptors that senses opioids are quite prevalent all the way from your spinal cord, actually from your sensory nerves all the way to the brain. There are opioid receptors, throughout the entire pain. People say the entire circuitry that is involved in pain, all of them express opioid receptors, which is one of the reasons why it's really hard to figure out which part of the pathway is contributing what. But one of the questions that you want to address is, does this context-associated tolerance, is it caused by spinal cord, or is it caused by something that happens in the brain? So to address that, these researchers had two conditions. One condition is they infused the morphine into the spinal cord. So this is kind of similar to the epidural that people might get during childbirth or pain management. And this would numb your pain kind of in your trunk body without going to your brain. And in those mice, they found that this context-associated tolerance does not develop. Whereas if in a second condition, they infuse the morphine directly into the brain, and the researcher showed that in this case, you can see context-associated morphine tolerance. So it tells you that it is actually happening, something is happening within the brain, this learned pain tolerance. So it's well and good to know that it happens in the brain, but the brain is a huge place. Where in the brain is it? It's actually scarily huge, even for a tiny little mouse. So what they did is they had so to answer that, what they did is they took advantage of this technique that we often talk about, which is immediate early gene. So if they had two groups of mice, one group had repeated morphine in the home cage, so those mice did not develop this context-associated tolerance. The second group of mice, the experimental group, got morphine in this new distinct context. So those guys did develop an accelerated, faster morphine tolerance. And after this has happened, they presumably gave one more dose of morphine and then euthanized the mice and then looked at the brain. And if an area has been active right before you euthanize the mouse, those neurons tend to make this immediate early gene that you can label for. They actually make several immediate early genes that you can look at, but one of the most used one is this thing called FOS, which is a transcription factor. Other immediate early gene includes Jason's beloved ARC, which is more involved in plasticity, perhaps. So think of it as a kind of like an fMRI. You want to see which bit of the brain lights up. The downside it is single-use fMRI. The mice are euthanized. You can't go back. So when they looked at, compared these two groups and looked at which bit of the brain is preferentially lit up in the mice that learned this context-associated morphine tolerance, they found three regions with increased FOS activity. The first one is this region called the dorsal medial prefrontal cortex. So it is the bit of the brain that kind of sits right above your eyes. And people think it's involved in working memory and kind of hand-wavy executive functions. Another area is this area called the basolateral amygdala, which is part of an area that people think is involved in valence, in things like fear, but also is something scary or also is something good. So kind of thumbs up, thumbs down of a certain environmental stimulus would involve basolateral amygdala being activated.
And we know a lot about that particular brain region in the context of the contextual conditioning
for fear.
That nucleus of the amygdala especially is a lot of the inputs that come in to the brain are going in into the amygdala and the hippocampus. So we probably would have predicted that if there's some sort of contextual information that's coming in, that it's either the amygdala or the hippocampus that's going to be important.
Exactly. So it makes sense. And as Jason alluded to, the third region that they did find is the hippocampus. The hippocampus also lit up in the mice that, got exposed to this context and also morphine.
And one can think of the morphine, the effects initially, that, as a human, if you take them, this is a massive stimulus to your everyday life, right? Obviously, sure. It's something that it's a big signal to the brain because it's such it's a big experience. So I guess it's not too surprising that it would make the mouse, associate this feeling or whatever the drug is doing to it with that context, because it's a big event in the animal's life.
Yep. But what's interesting is that the control group they're comparing to also got the same amount of morphine. So they also had this huge hit of, whoa, what is this? But the difference is mainly in these three regions that they found.
And what's you would probably expect if you did this experiment after that first day that there wouldn't be any difference in expression.
Exactly, because it is gradually learned.
Once you've done this, as you've alluded to, there's probably some extinction event that's happening where on the fifth day, it's the same thing. Like if you go, to your office and there's a surprise in your office, you're not going to be surprised after the fifth time you see it.
Right, exactly. What's actually quite interesting, quick comment, is that in the group that learned this context associated morphine tolerance, or as I say, I should use the technical term that they use, which is associative opioid analgesic tolerance, which is also a mouthful, but basically learned morphine tolerance. In the mice that learned it, when you give morphine and you put them back in the context. So as we will see later on in the later part of this paper, morphine actually works to suppress neuronal firing. It is coupled to receptors that actually makes the cell kind of less excitable. But in these mice, the brain is actually, these regions are actually activated. The brain is working hard to give you this tolerance, to clamp down morphine's effect. So it's actually your brain actively telling your body to ignore the morphine. You still want to sense the pain. So that's kind of interesting. So these authors have identified these three brain regions. Let me recount them again. The hippocampus, the dorsal medial prefrontal cortex, and also the basolateral amygdala. Three kind of very different brain regions and located in very different places.
I would say that, the amygdala and hippocampus is not too surprising. Also, prefrontal cortex probably not surprising either, in that in humans at least, and there's a lot of controversy or debate of how much the prefrontal cortex in a mouse is the same as a human, but in humans, it's thought that prefrontal cortex is a very powerful region to inhibit behavior. And ways think about compulsive behaviors, or in society we're not allowed to do X, and the brain region that says no, don't do your instinctual behavior, you can control it, is thought to be the prefrontal cortex, which is also why we think that teenagers lack that ability to block their impulsive behaviors, because the prefrontal cortex takes so long to develop.
Yeah, it's the last to develop, as I heard. Maybe in my case, it hasn't still developed. Who knows? But yeah, so these three brain regions are now, since they lit up in the mice that learned this morphine tolerance, the next question is, well, how do they talk to each other? So these researchers did a very nice experiment to try to tease that apart. What they did was they compared the effect in different groups of mice. They killed each of these brain regions, and they asked the question, how did it affect this learned morphine tolerance? In addition, how did it affect this FOS activation, this brain lighting up? So what they found was this: when they can kill specific brain region by actually injecting a tiny bit of virus, enough to cover the specific brain region, you hope, just that brain region and nothing else. They injected a virus that encodes a caspase-3 protein. And caspase-3, when it's expressed by a cell, triggers apoptosis, which is self-destruction.
Cell death.
Program cell death. Thank you.
Program cell death. It's literally killing the cells. So that's just a big caveat here, is that, you know, you're not silencing them, you're literally killing them.
Right. Yeah, there's no going back. So when these researchers killed the hippocampus, what they found was that it blocked this learned morphine tolerance. So the mice are now more the morphine now works better, even in the mice that were put in the new distinct context. So it blocked morphine tolerance. And what's interesting, it is also completely blocked the increase in FOS in the dorsal medial prefrontal cortex and in the basolateral amygdala. So both the prefrontal cortex and the amygdala effect is blocked. When they killed, or technical term is lesion, when they lesioned the prefrontal cortex, it also blocked this learned morphine tolerance, and it blocked the FOS increase in the amygdala, but it didn't block the FOS increase in the hippocampus. So immediately you kind of think, well, since the killing the hippocampus blocked everything, but killing the prefrontal cortex didn't block the hippocampus, what makes sense is that maybe the hippocampus is upstream of the prefrontal cortex, so that if you kill and the prefrontal cortex is downstream of the hippocampus, such that if you kill the prefrontal cortex, the hippocampus still gets activated because it doesn't affect the upstream guys. And finally, when they lesioned the basolateral amygdala, what they found was that it completely abolished morphine analgesia. So you don't even get any morphine analgesia on day one, or if you get any, it's severely reduced. But it did not block any of the FOS activation in either the hippocampus or the prefrontal cortex. So this really suggests that the kind of like the final hub is the basolateral amygdala. If you kill it, you'd block all the behavior effect of morphine, but you don't block any of the FOS activation of the region upstream of it. So based on this result, the authors proposed a model that when the mice are learning this context-associated morphine tolerance, the first thing that gets activated is the hippocampus. The hippocampus then talks to the dorsal medial prefrontal cortex. The dorsal medial prefrontal cortex then talks to the basolateral amygdala. And then the basolateral amygdala, through some unknown mechanism, mediates this analgesia or the tolerance, kind of opposite side of the same coin. So that's the model that they worked with. So it's good to come up with a model, but then you have to really test it. So the remaining of the paper that I'm going to try to go very quickly on is these researchers really trying to drill down these three brain regions. Do they talk to each other in the way that the model describes? And the first stop is, does the hippocampus talk to the prefrontal cortex? To show whether the hippocampus talks to the prefrontal cortex, what these researchers did was they injected a virus that encodes this protein called channelrhodopsin into the hippocampus. And channelrhodopsin is a light-sensitive opsin. So when you shine a light onto these cells that express channelrhodopsin, they would fire. And if you do it to the cell, these neuronal terminals that express channelrhodopsins, then the neuronal terminals would release whatever synaptic vesicles that they have. So what these researchers did was they injected virus that makes channelrhodopsin into the hippocampus. And then a few weeks later, they recorded in slice cells in the prefrontal cortex. And now they can shine a light and see, does the prefrontal cortex cell that they're responding from actually respond? Because if the hippocampus talks to the prefrontal cortex, then when you make the hippocampal cells fire using the light, it should release glutamate because these cells are glutamatergic. And glutamate will be detected postsynaptically if you record from the prefrontal cortex. And they showed that it actually worked. So,
and I'd say that's not new. I mean, we've known that for many years.
Okay. Thank you. Thanks, Jason.
So there's lots of talk from, even in memory consolidation, there's this idea that the prefrontal cortex is receiving input from the hippocampus to consolidate just generally memory.
Okay. They should have, in each figure, authors should have new or not new next to it. So that we know. So they confirmed that the hippocampus does talk to the prefrontal cortex. But the next experiment, what they did was they injected, so this is a little bit interesting, quite an interesting experiment. There are certain viruses that people have engineered so that if you inject into a brain region, it doesn't just infect locally in the brain region that you inject it into, but those viruses can actually get taken up by the terminals and then get transported all the way up the axon back to a different region that sends axon to the region that you just injected your virus into. So it's a little bit tricky to describe on a podcast, but it allows you to inject in region A, and this virus will get taken up, and all the brain region that talks to region A and sends axon to region A would also have that virus expressed. So it's a way for you to do this thing called retrograde tracing, where you can basically inject virus into a region, and then you can ask which other brain region talks to this place. So these researchers injected this retrograde virus into the prefrontal cortex. And lo and behold, this virus encodes a GFP, green fluorescent protein, so that it glows green. And lo and behold, you can see green cells in the hippocampus. It tells you that the hippocampus, further confirming, hippocampus talks to the PFC. But now it allows the researchers to record specifically from the cells that talk to the, specifically record the hippocampal cells that send projections to the PFC. So this is a very neat way of identifying circuitry. And what they found was, before they do the recording, if they train the mice on the morphine kind of learn context-associated morphine tolerance, so the mice that has been paired with morphine and this distinct context for several days, so morphine doesn't work anymore in these mice. If you then record for the cells in the hippocampus, the cells that project to the PFC, the prefrontal cortex, those cells are more excitable. So what that tells you is that learning this context-associated morphine tolerance actually make these hippocampal cells that talk to the PFC more ready to fire. And that makes sense. That kind of is in agreement with increased FOS that you see. Like those cells are more ready to fire. And therefore, if you put the mouse back in the context and give it morphine, those cells do fire. And so that tells you that those hippocampal cells are, associated, their increased activity is associated with your learned morphine tolerance. And what the researchers then did was, right now, up to this point, we only know that the hippocampus, increase in hippocampal firing is associated with this morphine tolerance. We don't have any way of perturbing it yet, so that we don't have a causal mechanism. So in order to do that, what you really want to do is actually inhibit these cells when you're pairing the context with morphine. So let's say if you block these cells using a kind of a designer engineered receptors. We won't have time to go into that here, but we can specifically make these cells to fire less by giving a designer drug that matches a designer receptor that we express only in these cells. If you selectively shut down these cells during the 5 days when you're giving morphine to the mice in this distinct cage, then you can block this development of learned morphine tolerance. Conversely, if you put in an engineered receptor into these hippocampal cells that make the cells fire more, so now you artificially boost these cells and make them hyperactive, then if you do that, then give mice morphine and just have them in the home cage. So remember, these are the control mice that previously did not have this context-associated morphine tolerance. But if you artificially increase hippocampal cells firing, specifically these cells that talk to the medial prefrontal cortex, then these cells, these mice actually have a much faster morphine tolerance, as if they have learned the context-associated morphine tolerance. Presumably, it's not specific to any context because the mouse didn't get put to a new context, but in a sense, the mice have a stronger morphine tolerance. So it is all in agreement with activating the circuitry, leading to the development of morphine tolerance. But what's interesting is the researchers quickly asked the question, well, we know now, based on all these data we've got, we know that hippocampus talking to the prefrontal cortex is important for the analgesic side of morphine and the tolerance. How about the breathing side of morphine? So morphine also suppresses breathing. Do you see breathing suppression if you pair it consistently? Do you see tolerance to morphine's breathing suppression effect if you pair morphine injection consistently to a new context? And the researchers show that, yes, you do. You see an accelerated development of tolerance if you consistently pair this new context with morphine. So it's also learned. This is kind of scary and potentially involving the brain. But what these researchers show that if you selectively inhibit the hippocampal cells that talk to the medial prefrontal cortex, if you inhibit those cells, while it can block the development of morphine tolerance for pain-killing effect, so for the analgesic effect, it didn't actually block the development of tolerance for morphine's breathing suppression effect. So it suggests the hippocampus talking to the prefrontal cortex is not involved in this learned tolerance for breathing. But what's interesting is that because it is still context dependent for this breathing suppression, it presumably still involves the hippocampus. And what we don't know is whether it is actually the hippocampus talking to another brain region that's important. So that's an open question.
So Tim, what the reason that people overdose is because in the home context they develop tolerance, and then they use increasing doses, and then when they go to a different environment, then there's no longer tolerance with respect to respiratory function. Is that correct?
Right. One of the hypotheses. People think it might contribute. Yeah. But it's interesting because in the experiment we've been talking about so far, the control group also got the morphine at home, but they don't seem to develop this tolerance. And the reason is because whereas in human, if you're using, often when you're using opioid drugs at home, you are using it pretty much 24/7 to ward off the effect of opioid withdrawal, which is nasty, cold turkey, etc. Whereas in the control group in this experiment, the mice receive morphine at home, but they only receive it for one brief moment during the day, and then they probably go through extinction and they go through withdrawal. So that might be the reason why. So it's a slight difference. Yeah. So next, the researchers, so far we have established hippocampus talking to the prefrontal cortex is important. The next route of this relay is the prefrontal cortex talking to the basolateral amygdala. So the researchers then looked at this part of the circuitry, and they kind of did a very similar experiment where they injected these designer receptors that can either make the cells increase or decrease firing into the prefrontal cortex. But these receptors these designer receptors are engineered so that they are inert. They cannot even be expressed unless it sees a second signal, a kind of molecular scissors that you have to put in a separate virus. And these researchers put in the kind of these scissors protein virus into the basolateral amygdala, which is the downstream region for the prefrontal cortex. And this virus is a retrograde virus, so it would be taken up by the axons and get transported back to the prefrontal cortex. So now this allows you to, is a very clever way of allowing you to really probe of all the cells in the prefrontal cortex. I want to either activate or suppress only those that talk to the basolateral amygdala. So when they did this experiment and when they artificially inhibited these cells from the prefrontal cortex to the basolateral amygdala, they were able to block the development of this context-associated morphine tolerance. And conversely, when they artificially boosted these cells' activity, they were able to increase this morphine tolerance even in mice that were in the home cage. So it's as if they were paired with a context. Mm-hmm. And the researchers also did a bunch of experiments to look at the kind of online activity of the prefrontal cortex cells using calcium imaging. So what they did was they put in a little lens in mice that are actually alive and moving, and so that they can look at calcium activity. So these are cells that have been engineered to glow more brightly when when the cells are firing more, so that there's more calcium inside the cell. And they found that in cells that have learned this morphine-associated analgesic tolerance, when you put them into the correct context, then the dorsal medial prefrontal cortex fires like crazy. But if you put them in the home cage where they didn't get morphine, the prefrontal cortex don't really fire. And they don't see this difference in the other groups. So it really tells you that the prefrontal cortex talking to the basolateral amygdala is important. And
this paper uses all the new modern techniques in neuroscience.
This paper
manipulating neurons in either with light or with these chemical biology approaches, imaging in vivo. Yeah, it's nice.
Yeah, they really did a lot of crossing T's and dotting I's, which I like. So the researchers were further able to show that in the dorsal medial prefrontal cortex. So this middle relay station in between hippocampus and the amygdala, these cells that talk to the amygdala express this other peptide called cholecystokinin, CCK. And CCK, cholecystokinin, has previously been shown. It's actually a protein first identified in the gut. Cholecystokinin translate to bile sac moving. So it actually causes your bile in your gallbladder to get secreted. So it's a neuropeptide for digestion, but a lot of peptide out in your peripheral body is also used by the brain for communication. So this cholecystokinin, CCK, is actually, turns out it is being released by the prefrontal cortex neurons that project to the amygdala. So they were able to show that these cells actually express CCK. And it's important because previously people have found that if you inject CCK agonists, so drugs that activate the CCK receptor, you can actually counter the analgesic effect of morphines. So it's building a picture that what might be going on when the mouse is put back in a context previously associated with morphine is that your brain is now in your prefrontal cortex activated and releasing CCK in the basolateral amygdala. This is what the researchers wanted to show in the final figure. And what they did was they activated the cells in the prefrontal cortex. Actually, first of all, what they did was they just infused CCK blocker or agonist directly into the basolateral amygdala. So very brief background is that CCK receptor, if you activate CCK receptor, the cells tend to fire more. It is an excitatory receptor, and so that is an agreement with the BLA being more active when the mice are undergoing this learned morphine tolerance. So what they found is that if they infuse a CCK agonist, so drug that activates a CCK receptor, directly into the basolateral amygdala, then in mice that receive morphine in the home cage, so mice that normally would show very slow tolerance, the tolerance is sped up. So kind of similar to the behavioral effect of context-associated morphine tolerance. In contrast, if you block the CCK receptor in the amygdala, in mice that have morphine paired with this specific distinct context, then you can block this learned morphine tolerance. So further tells you that the CCK is very important. CCK receptor in the basolateral amygdala is very important. And the researchers further show in slice recording that actually if you record from a basolateral amygdala cell, so this final pathway where all this analgesia and also tolerance to analgesia is we think is kind of piping through. If you record from an amygdala cell, if you put in a mu opioid receptor, so kind of like a morphine analog, those cells would decrease its firing. So they would quiet down. Whereas if you put in this quieter down, so whereas if you add the morphine or morphine analog plus a CCK agonist, so something that activates the CCK receptor, you can undo morphine's effect. So now the cells is firing more. So this really suggests that morphine and CCK are kind of countering each other. Morphine is trying to suppress your basolateral amygdala. CCK is trying to activate it. And what might be happening is, as you put the mouse back into a previously learned context that's been associated with repeated morphine, the CCK positive cells is hyperactive, releasing CCK into basolateral amygdala. And this causes the basolateral amygdala to be hyperactive and kind of blocks the effect of morphine. So I think that is, I know Jason has to go, so I'm going to wrap up there.
Well, I think it's, those are that we sort of got into the weeds a little bit there about the details of this pathway. But I think what it brings up is that, as you mentioned in the beginning, is this idea that tolerance is not set; it's malleable, depending on the environment that you're in is important. And it seems like it's something that had been, I don't know, forgotten, at least in the literature for a while. So, I think this paper may hopefully revisit the sort of behavioral aspect of it, but also now provides a clinical target that you could maybe use to prevent or alleviate tolerance, which seems to be the biggest reason why people overdose, at least.
Yeah.
How would that work, Jason? What are you thinking about?
Yeah, I think in practice it's going to be difficult, right? You'd have to basically have a drug that people would take at the same time as the morphine, and
to avoid overdose.
To avoid overdose, and I think that seems in principle doable. I think it just depends on what the other side effects of that drug are, and how specific can you get, and is it still, is the analgesia still enough? So getting that dosage right is probably very difficult.
Yeah. You can imagine.
So it sounds like it could also encourage more abuse, right?
Yep.
Yeah.
But we don't know how separate the pain-killing effect of morphine versus the breathing suppression of morphine versus the euphoria effect of morphine.
Yeah.
They might very well be mediated by completely different brain pathways. So we know, for example, that the targeting the hippocampus, talking to the prefrontal cortex, blocked the analgesic part, but not the breathing part. So we don't even know whether, let's say, you want to block CCKs, this CCK receptor, so that the person using opiate would still sense some of the effect of morphine, but maybe that's only for the analgesic, but not for the breathing suppression. And that would be, not good enough.
Yeah, exactly. And also, I think it's interesting that the amygdala seems so important for the expression of the analgesic effect.
Yeah.
I don't know if that's been known before, but
that's actually quite surprising to me that you can completely block it. What's actually a very final kind of in the weed point that they don't talk about in the paper, but what's quite interesting is that they showed that you can put in morphine directly into the basolateral amygdala, and that would give you analgesia straight away. And we also know that if you give CCK into the basal amygdala, you can counteract the effect, at least inside a cell. You can counteract the neuronal suppression effect of morphine if you give CCK agonist. But behaviorally, CCK agonist didn't work on the very first day. It actually, you have to learn it before CCK agonist actually does anything. So there's some interesting learning plasticity going on, maybe in the amygdala, maybe elsewhere, that is important. That is still needs to be, found out.
Yeah. Cool.
That was great. Thank you, Tim. That's just an awesome paper. I love it.
It's pain is very interesting. There's actually, I didn't get to talk about it, but it's possible that the non-context specific tolerance, so like so mice in home cage with morphine also showed some morphine also worked increasingly less well. And there are papers kind of somewhat recently published that suggest that might be due to spinal cord or kind of peripheral nerve mechanism. So there are different things that might dissociate it differently inside the brain. And it's kind of, scientists have to figure out. But hopefully, we just basically want a painkiller that has no abuse potential and no breathing suppression effect. Yeah.
All right. Show notes for TWiN43, microbe.tv/TWiN. Questions, comments, twin@microbe.tv. Your support, we'd love to have it. Microbe.tv/contribute. Jason Shepard's at the University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Tim. That was nice. That sort of combined my interest in associative learning with your interest in pain.
Tim Chung is at New York University. Thanks, Tim.
Thanks, everyone. That was fun.
I'm Vincent Racaniello. You can find me at microbe.tv. You're listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.